Hello, everyone. I hope you can um, see the slides and hear me properly. So, welcome to the session 7 of the NPTEL Cell Biology Cellular Organization, Division, and Processes. This is a course that is instructed by Professor Shrikala Loria from ISC Bangalore, and I am one of the PMRF TAs of this course. I am Smriti. I am a PhD student from ISC Bangalore. So this discussion session is basically a doubt clearance session. If you have anything that you want to discuss or ask, you can go ahead. But apart from that, I would be discussing the concepts that have been taken up in this week of the NPTEL course and discuss the previous year assignment questions. So when we just look at these two images, these are microscopic images of two different cellular population. Let us try to identify what exactly is different in these two cells. So on the outlook, the first thing that you notice is that the cells on the right have taken up more of the dye than compared to the cells on the left. Now, this is a beta galactosidase activity screening, in which what they basically do, it's similar to the blue and white assay that is used for bacterial colony screening. Here, they supply the cells with XGAL, which is a IPTG analog, and that when activity is there, it is when it is cleaved, then it becomes blue in color, which we can observe. So here we can first simply say that when compared to the cells on the left, the cells on the right have taken up more of the dye. Now, also you can notice that the cells are bigger and fatter when compared to these set of cells. And if you notice that there are some cells with a double nucleus, like this cell has two nucleus, this cell has two nucleus. So there are multinucleated forms as well. And in general, cells have grown in size and are bigger. So why is this? What are the two populations of cell? So what they have tried to represent here is a normal cell and a senescent population of cell. So what happens is senescence is set in. It is a non-reversible replicative senescence, which means that these cells will never be able to replicate ever again. So now why this happens is because these cells have accumulated some sort of damage. It may be DNA damage, it may be chromosomal organization damage, or it may be chromatin organization damage. And because of this, the cell recognizes this damage and halts. It does not want its progeny to inherit any damaged uh, information. So they are senescent. They are there are certain characteristics of these senescent cell populations that is used to differentiate between them in a experimental setup. One is that it's morphologically and metabolically altered. So in this case, it has more of the beta galactosidase activity. This is known as senescence associated beta galactosidase activity. This is mainly because there is a larger accumulation of lysosomes within these cells. And here, the beta galactosidase, which is an enzyme that is important in carbon metabolism, is more active. There is also chromatin reorganization. There is some aberrant chromatin formation. Like I mentioned, that there might be two nucleus in some cell. The nuclear envelope may be disrupted and stuff like that. In general, there is a chromatin improper chromatin organization that is seen. There is also altered gene expression levels. Some proteins are are greater present, and because of this, there is a predominantly pro-inflammatory phenotype. You would have heard that there are two types of inflammatory signal, pro and anti-inflammatory. They are characterized by a large number of interferons, large amount of cytokines and stuff like that. And we see that the senescent cells have a predominantly greater pro-inflammatory phenotype, which means that they are more actively synthesizing pro-inflammatory cytokines, and this secretion is known as the senescence associated secretory phenotype. Because otherwise, for the synthesis of certain protein, synthesis of such proteins, you require certain signal, require certain stimuli. But in these cells, just the triggering of senescence itself is enough for it to attain this phenotype of senescence associated secretion. So these are the characteristic features. Any one of this can be used to experimentally differentiate between cells. And in this slide, I have just tried to create a representation of the different techniques that you can use to identify which cell in your population is having the senescence phenotype. So in this first image, the image on the left, here the green signal that you've seen is the staining of lamin. So if you can recall, lamin are these proteins that are present on the nuclear envelope. Their phosphorylation, when they're hyperphosphorylated, then there is nuclear envelope breakdown. And then when they have to reassemble, then their phosphorylation is lost and they reassemble to form a network, a mesh-like network, which stabilizes the nuclear envelope. So it is seen that because of loss of chromatin organization, many of these cells have loss of lamin. Here we can see that these cells in the middle, they have completely lost the lamin signal. And even there are other groups of cells which have a very low intensity of lamin. Maybe they are just initiating their uh, loss of lamin. Here in the second image, the image in the middle, this is a stain for phosphorylated histone H2A. So the green signal is that of phosphorylated histone. So in senescence phenotype, apart from improper chromatin organization, there is also a trend in which you can see that the entire genome, their epigenetic marks have changed. So in the previous sessions, we have seen that heterochromatin and euchromatin are two different types of packaging. Heterochromatin is more tightly packed and there is silencing of the genes that is present in that region, whereas euchromatin is the active genes that are being transcribed and translated into proteins. So apart from that, these histone molecules that form the nucleosome, so there is a DNA that is wrapped around these histone molecules to form the beads on string structure. So these nucleosome have histone 2A, 2B, histone 3 and histone 4. And then in between, there are linker histones, which are histone 1. Now, this nucleosomal core, which is the histone octamer, has multiple modifications that is seen, including in some cases phosphorylation. The most common ones that we see are methylation and acetylation. So acetylation imparts this negative charge, because of which it can be brought close to each other, because of which it is spread out. That is the euchromatin arrangement. Heterochromatin arrangement, they are tightly packed to form the 30 nanometer fiber, if you can recall what we have discussed previously. Similarly, phosphorylated histone, they have certain other implications. These will have implications for the gene that is present around the histone. They may be active, they may not be active based on the place in which it is phosphorylated. So it has been seen that in a senescence phenotype, there is a greater amount of presence of phosphorylated histone H2A also. This we'll know when we compare normal cells and senescent cells, the signal will be much greater. Again, in this third image, HP1 is a protein that helps in the progression of heterochromatin. So what happens is there are, like we discussed, chromatin readers and chromatin writers. So in this case, when HP1 first recognizes a mark that is supposed to be repressive, which means the gene has to be silenced, it will first condense that part, as in more tightly pack it, and then go on, and it goes further on and tries to pack that entire region into a heterochromatin. This progression of heterochromatin is mediated by this protein known as HP1. So it is also seen that in a senescent cell, there is a greater amount of HP1, suggesting that globally in the cell, there is a greater silencing of the genes present, which means that there is a, in general, lower expression levels of various genes, because the cell is slowly aging and is destined to die. So, however, we have previously discussed that senescent cells have completely lost their replicative capacity, which means that they're uh, irreversibly they can never replicate ever again. So why can't we simply use that as a marker? So we have seen previously that there are BRDU staining assays, there are BRDU incorporation, EDO incorporation assays that will help us identify whether a cell is going to replicate or not. Why can't we simply use that as a marker? So that is very tricky, because there are different types of um, cells in which there is a cell cycle arrest, which may or may not be reversible. So the three type of cells that is depicted here are senescent cells, quiescent cells, and differentiated cells. So in senescent cells, we've seen that it's an irreversible arrest, And this is because of some macromolecular damage. There are certain specific signaling pathways that are upregulated, which result in the senescence associated phenotype, and there is active secretion from the cells. However, in a quiescent cell, it is a reversible arrest. There is no damage to the cell as such, but the reason it is not replicating is probably because there is no available nutrients, or there are certain contact inhibition signals that tell the cells ki no, you should not replicate right now. And this is a reversible arrest. When the situation is more favorable, it will again go into cell cycle. Then there is a third type of cell known as the differentiated. They are terminally differentiated cells. They also have irreversible cell cycle arrest, but this is not because of macromolecular damage, but because of certain signaling pathways that help it arrange its chromosomes and chromatin in such a way that only a certain set of genes are turned on, and only those genes are able to produce proteins and thereby have their effector functions. Remaining set of genes are turned off and they are not expressed in these cells. So different type of differentiated cells here would be your liver cells, your stomach cells, your epithelial cells, your fibroblasts. They are all different terminally differentiated cells. However, senescent cells are those that have reached a potential of damage beyond which, if it replicates, then the progeny will inherit some mistake, because of which the cell is stopped from replicating for the entire organism's benefit. So, how was this identified? How was cell senescence identified? It was thanks to this researcher Mr. Hayflick, who first proposed something known as the Hayflick's limit. So if you think about it, there was a time where people did not know that, okay, senescence will be reached, the cell will stop dividing at one point of time or the other. And in fact, August Weismann in 1881 first suggested that death takes place because a worn out tissue cannot forever renew itself, and become a capacity for increase by means of cell division is not everlasting but finite, which means that there are a lot of mistakes that happen in your cells. There is DNA damage, mutation with every cell cycle, and there are DNA repair pathways also to uh, make it right, to correct these mistakes. But according to Mr. August Weismann, he suggested that actually senescence, aging, and death is because the cell has lost its correcting ability. And that is why it is not going to forever renew itself, and cell division is at one point of time going to stop. However, there was this scientist in 1921. Uh, he refuted this argument by saying that all cells explanted in culture are immortal, and that the lack of continuous cell replication is due to ignorance on how best to cultivate the cells. So, explant means you take it out of an organism, put it in a dish, and try to culture it. This is known as explanting. So he suggested that if he takes chick heart fibroblasts, that is fibroblasts from the heart of a chick, it can continuously grow for 34 years, and he experimentally showed it. However, what later came into the light is that he used to feed these chick fibroblasts with fresh fetal fibroblasts. So it was basically just a fetal extract that he fed to these cells. However, that extract also had fetal fibroblasts present in it, and it is probably that kept rejuvenating, and it was not the original culture that grew continuously for 34 years, but newer cells that were added to the mixture, that is what continued to grow for 34 years. So this came into light when the scientist, Mr. Hayflick, he performed an experiment. He wanted to grow fibroblasts for a longer period of time, because it is very tedious to isolate fibroblasts every time in order to conduct an experiment. So in order to avoid that, he wanted to have a stable culture. He wanted to grow it for over 1-2 years continuously, and he saw somehow that every time after a specific amount of cell division, the cells died. So here is a representation of his fibroblasts. These are young fibroblasts and these are old fibroblasts, and you can clearly see the morphological difference. You can see that the cells have become larger, they have different morphology, they are more scattered, and they don't look healthy, right? So he saw that after a finite number of division, these cells inevitably, no matter how careful you are, no matter how carefully you keep changing the media, you keep the cells in the right environment, they die. So this he estimated that there are three phases of cell culture. The first phase is the stationary phase. It's very similar to the bacterial growth curve. Stationary phase where the cell is still taking time to divide, the number of hours it needs to divide is very high. And then there is a log phase where the cell is continuously growing and dividing quickly to double its population. And ultimately, after a certain number of divisions, it ultimately reaches senescence, after which it does not divide and ultimately dies. So he conclusively proved this by performing an experiment wherein he took fibroblasts from male mice, grew it for a certain amount of time, and then later to the same culture he added fibroblasts from female mice, and then continued to grow it. After a greater amount of time, when you sampled these cells and then checked if they were from the male or the female mice, he inevitably saw that the younger cells, that is the cells taken from female mice, survive better when compared to the older cells, that is cells taken from the male mice. This suggested that it is not because of anybody's care or uh, any specific condition, but it is because of a inevitable uh, accumulation of DNA damage, which leads to senescence in a cell population. So with this limit, that is the number of times a cell can divide before which it attains senescence, that limit is known as the Hayflick's limit. Now he proposed that this is because of the telomere shortening. Now we have seen previously that excuse me, telomere is the ends of chromosomes that are repetitive sequences of DNA that are mainly present only to avoid the end replication problem. So again, the end replication problem arises when there is a DNA polymerase, but it cannot de novo generate a strand. It needs the attachment of primer, and it can only synthesize DNA from the 5' to 3' direction. So there is this one primer that is attached to the 3' end of the DNA, which is not completely, like when it is removed, it cannot be completely regenerated. Because of that, every time a DNA replicates, there is concomitant shortening of the ends of the chromosome. This is known as the telomere shortening. Now, this is often overcome by this enzyme telomerase, which is able to add these repetitive sequences to the end of the chromosomes, ensuring that essential information from the DNA is not lost. However, as the cell ages, it is known that the telomerase activity keeps on reducing, because of which ultimate there is a critical shortening of the telomere. This is known as the critical length of telomere, beyond which it will simply not divide and enter senescence, and that number of division is known as the Hayflick's limit. Now, moving on, we've spoken about uh, primary cell culture, wherein you take cells from an organism and grow it in an in vitro condition. How this is actually done is here, this representative image is the isolation of peritoneal cells from mice. So peritoneal, you would have heard before, is a cavity that encompasses all your abdominal organs. So if you have to isolate peritoneal cells, there is a greater presence of macrophages in the cells, and they can be used for studies that require these cells. So what they do is basically they first they add certain irritants to the peritoneal cavity that will enhance the accumulation of macrophages in the peritoneal cavity. More and more macrophages will be recruited to the peritoneum, and now you can isolate it. So then you cut open the mice, you slowly cut open the skin of the mice, ensuring that the peritoneal cavity is not nicked, and then you flush the cavity with PBS, and then you take out the fluid. So this fluid will be a mixture of cells. It will also have a lot of RBC present in it, and that can be pelleted down. And then later add RBC lysis buffer, which will get rid of all the RBCs. And now you'll see a white pellet. Now the red pellet signifies there's RB contam, but after RBC lysis, you'll see that there is a white pellet. The white pellet can be resuspended in the media and put into these dishes. These dishes are known as tissue culture dishes, wherein you add the appropriate media and allow these cells to grow. However, these cells, like Hayflick identified, will only survive for a particular amount of time, after which it will not be able to survive. However, we have commonly heard of these cell lines, HeLa cell line or THP1 cell line, and these cell lines are immortalized, which means that they will never die. No matter how many times you keep on passaging them, keep on splitting them into fresh culture, it just keeps on dividing, and dividing. It never stops division. This is known as an immortalized cell line. And there are many commercially available immortalized cell lines. There are lung cells, there are epithelial cells, fibroblast cells, kidney cells, there is a macrophage cell line, there are lymphomas, there are based on what exactly your question is, what model system you want. There are a lot of different types of cell lines available. Now, how these immortal cell lines are available is that most of them have been transfected with some virus or have a cancerous property, because of which they have lost this control. They keep dividing no matter what happens. They have a high telomerase activity and their telomere never shortens, and they just keep on dividing. This is very useful for any cell biologist, in fact, any infectious biologist also, because imagine you have a drug and just COVID 2, SARS-CoV 2 only, right? There was SARS-CoV 2, and there was an active uh, research going on for the hunt of the proper vaccination. Now, say all these vaccine had to be treated on humans or tested on humans, then you need to sign these ethical clearances. You need to say, okay, I am ready for the consequence. It can even be fatal, right? You can't just take and just test it on humans. So then you need animal models, and that requires a lot of animals. So if you have to test each and every vaccine, each and every compound on animals, that again raises a lot of ethical issues, because animals is animal testing is also very well regulated in the world. So the solution is to test them on cells, because they are easy to get and they're easy to infect. You just have to infect them with SARS-CoV-2 and then later test if the drug is working on it or not. And if it's a vaccine, then vaccinate the mice, isolate the cells. So that way, you're not sacrificing mice for one compound. You can take its cells and again use them for different compounds. So that is why cell culture is very important. But however, if you have to perform all toxicity studies on mice or primary cells, then again isolating them each time is a very tedious process, and they won't survive for long. They will probably die within, say, a month or so. So, in order to avoid that, cell culture, when it was first established, was a big advantage to all these scientists, because now they just needed to thaw one vial that they already have, and then you can repassage them, keep on passaging them for infinite amount of times. Now, the very first immortalized cell line, I'm sure that anybody in biological research would have heard of, is known as HeLa. HeLa cells are a cervical cancer cell line. It was first isolated from this person known as Henrietta Lacks, and it was named after her. He for the first two letters of her first name, and La for the first two letters of her last name. Now, her story is very uh, sad, because she was i- affected with cancer at a time where racism was very much prevalent in the US. She was an African American woman, and most hospitals did not even treat people from that race at that particular point of time. So when she did go to a hospital, they had a special wing for people of a different race, and Caucasians were treated differently. So in that wing, uh, she was diagnosed with cervical cancer. There was a lump that was growing inside of her cervix, which was because of a human papilloma virus, HPV virus, which basically goes and inserts its DNA into the DNA of the host, and because of which certain genes are activated and certain genes are silenced, and cancer develops. It's one of the most common causes of cervical cancer. But however, in her case, she also had syphilis, because of which her immune system was immunocompromised. She was not very fit. Because of these two factors put in together, the cancerous cells were so potent and so fastly dividing that they provided the ideal starting point, because before that you don't know what media to grow them in, how long to grow them in, what conditions to grow them in, because the cells are very sensitive. They require the right amount of nutrients at the right amount of time. But such potent cells like HeLa cells, when they were isolated, before which there was no cell culture that was possible, they isolated a scientist known as George K. First put them in a culture and kept them in an incubator. And because he had done this hundreds of times, he had isolated clinical samples hundreds of times and done the same thing. And he did not expect them to survive after 2 days. But to his surprise, he took the flask out after 2 days and not only saw that the cells were alive, but they were also actively dividing. They had increased in number. And when you repassage them, they just kept on dividing, kept on dividing. And that is how cell lines and cell culture was born. So even though she died a very tragic death and her cells were taken without her permission, it has revolutionized biological research, and there are a lot of important events in which HeLa has been a crucial part of. Uh, to mention a few, it is the discovery of the polio vaccine, the identification of the role of HPV in cervical cancer, the actual study of telomerase and how telomere shortening affects cells, and also the mapping of human genome was also for the first time uh, done on HeLa cells. So, so uh, later there was a very big controversy that came out because they started extensively studying the genome of HeLa, and there were a lot of DNA hereditary based information about their family, about what diseases they're susceptible to, about where they come from, a lot of personal information that was made public. And then the family had raised issues, and there was a legal case that was brought up regarding the actual ownership of HeLa cells and what you should do with it. But apart from that, HeLa has played a very important role in HPV virus and vaccine, because again, HeLa, like I mentioned, was infected with HPV, and because of which it's easier to use those cells for the vaccine study, right? It's easier to use those cells for drug study also. Apart from that, polio vaccine, when polio was very prevalent, and I'm sure all of you would have gotten your polio drops, the oral polio vaccine, OPV that we call. But before that, when polio was very prevalent, and there were large amount of children dying of polio, and even those that survived had a lifelong paralysis, their limbs were not proper. And because of this, there was an urgent need for development of vaccine. And again, when monkey testing and mice testing were not very feasible, scientists used HeLa cells and they found out what is a ideal vaccine candidate, and they tested the toxicity and all in HeLa cells, and later on uh, uh, administered a large amount of children with this vaccine. This is not the oral polio vaccine. This is a vaccine that was an inactivated polio vaccine, IPV, in which the virus is inactivated and that serves as a vaccine. And that really reduced the amount of cases of polio that they were seen, and completely changed the way the world was dealing with polio. Again, human genome project we've heard of is when human genome has been sequenced and you know where what DNA is present and what chromosome. However, how you need to map it, right? You need to map that DNA to a particular protein, and you need to be key. Okay, this is the portion of the DNA that is encoding for this particular portion of the protein. That was actually done by like scrambling up the human genome. The human genome were mixed along with that of mice, and then randomly injected into cells. So there are certain regions in which the mice, the particular human genome is not present, the human gene is not present, and based on that you can find out okay, this protein is not being expressed, which means that this is because of this sequence in the DNA. So, moving on, we've spoken about tissue culture, and I just wanted to give you an overview of how tissue culture lab looks and what exactly we use and what is the media or what are the terms that we use in cell culture. And here, seen over here, is a typical cell culture hood. So, again, because cell culture can be very easily contaminated by bacteria, fungus, and so on, it is handled inside a biosafety cabinet. So inside the biosafety cabinet, you have appropriate PPE, you wear gloves, and the cells are grown in these dishes that are known as flasks. So there are different types of flasks. These are known as T neck flasks because they have a T like neck structure, and based on the area, so this is known as a T75 because it has 75 centimeters of area, and this is known as a T25, a smaller flask. So if you look at adherent cells, right, cells that require a substratum to attach to and then divide, they require surface area. So if you have a greater surface area, the greater the number of cells you get, and if you have a smaller surface area, the smaller the number of cells you get. These are dishes, cell culture dishes. The 100 dishes and smaller dishes are also available, and these are plates. So based on the number of wells, these plates are classified. This has 6 wells, so it's called a 6 well plate. This has 12 wells, so 12 well plate, 24 well plate, and this will have 96 wells. So this is known as a 96 well plate. Why this is required is for, say you have a compound. Simply, I'm just testing the toxicity of compound A. I will seed cells into these wells and at increasing concentration, supply them with A, compound A, 0.5, 1, 2, 3, 4 micromolar, so on. Then I will check the viability of cells. Which concentration did actually the cells die? What is the toxicity of this particular compound? So in that studies, 96 well plays a role. Similarly, if you just want to compare two conditions, okay, in one I'm supplying the cells with PBS and one I'm not. What is the difference? That you can simply use 6 well plates and based on your need, modulate the number of well plates that you used. The other thing that you see inside this hood is the presence of these media. Cell culture, like bacterial culture, have specific requirements of media, and this is a schematic as to what are the basic components a cell culture media requires. So again, if you just think about it, it requires nutrients, it requires carbon source. So there is an absolute need of carbon source, absolute need of metabolites like vitamins, minerals, amino acids. So there is also requirement of a buffering system because if you see your pH inside this body is very well maintained. It's known as a physiological pH, and the cells will have those function only in those pH because your enzymes are active in only that pH. So if you take any enzyme, it has an optimal pH curve, which means that even to the left or to the right, more acidic or more basic, it will not perform its maximum effect or function. Only in that particular pH will it have the proper functioning. So, uh, so there needs to be a buffering system in place in order to maintain that physiological pH in the cell culture. Again, if you've heard about it, cells are placed in a CO2 incubator, which means that it cannot be placed in a incubator that has 100% oxygen. That is toxic to the cells. So we always maintain a 5% CO2 level in the incubator. That is where cells will survive. So 5% CO2, that CO2 will be able to interact with the H2O present in your media, water present in the media, and generate H plus ion. So it will make it more and more acidic. So in order to prevent that, we generally add a buffering system. You add like buffers like HEPIs or sodium bicarbonate, which will prevent this pH from fluctuating so much and will maintain it in the range of 7.4 to 7.8. Now, again, you add antibiotics. This is very controversial. You may choose to or not choose to add antibiotics based on your application. This is basically to prevent bacterial contamination because cells are very easily contaminated by bacteria. There is again a pH indicator. Most common pH indicator is phenol red. It changes color, it becomes yellow when the pH is too acidic and becomes pink when the pH is too basic. So the optimal color of the media is this reddish orange color that will tell you if the nutrients are being deprived, then the pH will become acidic, it'll turn more and more yellow in color, and that is when you know, okay, you have to change the media out of these cells. There is also presence of serum. This acts like a, a bulk source of nutrients, vitamins, minerals, metal ions, and all the other essential factors that is required for cell culture. Again, there are serum-free media also available based on the application, because serum varies from batch to batch. It is just isolated from, so you get FBS, just fetal bovine serum. It's just isolated from the bovine, which is the cow's fetus. So because of that, it will vary from batch to batch. The exact amount of nutrients is not specified. So if that plays a role in the thing that you're trying to investigate, then it'll be a problem. So people tend to use serum-free media in that case, where they grow cells without serum. But of course, the cells will not survive for as long as it will with serum. So based on the application, a lot of this can be altered. Now, again, primary cells also can be immortalized. So, say you have to isolate a cell from this particular mice, and that is the same cell that you keep on want to studying to base your studies on. So, for that, you can insert um, a TERT enzyme, which is telomerase enzyme expression. So if you add telomerase enzyme under a constitutive promoter, then it'll keep on producing telomerase enzyme in a large amount quantity, and it will not allow telomere shortening, and because of that, a cell expansion is possible. So they can be put in a plasmid with the antibiotic selection, and then put the drug in order to select for the cells that have taken up this enzyme, this construct expressing the enzyme, and then those cells will be immortalized, and you can just make vials of them and store them in liquid nitrogen, thaw them when needed, and they will have this immortalized nature. They also perform viral transduction. So there are certain viruses that when infected in a cell, it has activates certain genes, because of which there is induction of cancer. They're called oncogenetic viruses. They induce cancerous property in cells. This is also used to immortalize cell lines, and many of the cell lines that's commonly in use. One such example is the raw 264.7 macrophage cell line that has been transducted with viruses, because of which it now has this immortalized property. So one such interesting application of these immortalized primary cell lines that I could find is something known as meat culturing. So, with increasing amount of awareness that global warming and CO2 emission levels is happening, it came to realization that to grow cattle, that requires large deforestation of forest areas in order to grow the feed that they will feed on, to grow grasses and grasslands and so on. So, there is wide deforestation and large amount of water consumption to grow those grasses that they eat, which means that there's high CO2 emission in order to grow even say 10 or 20 number of cows in a farm. So if you're growing them for meat, then by consuming the meat, you are contributing to so much of CO2 emission. So this is when people started getting aware there must be some alternative for this. They started establishing, and still a very new field where meat culturing is a thing. So here you take out cells from the animal, and then you isolate the desired cells. So you require fat cells, so you require muscle cells based on that, and then you immortalize them, and then you grow them in large quantities in bioreactors, harvest them, and finally seed them in specific proportion. This much fat to this much muscle, this much of other cells that they would probably require, and it will give you a product which is a cultured meat, which is a lab made meat, which has similar texture, taste, and cook similarly to that of the original tissue that is obtained from an animal. So because this does not involve rearing animals, this is a greener alternative to meat. And this is being increasingly popularized in various countries, and meat culture is an active area of research. Now, scientists are trying to identify that, because now you can group a bunch of cells and have minced meat that is very easy to make, but still, in order to give it a structure like steaks, or to give it a structure like chicken piece and so on and so forth, there needs to be some scaffold within which these cells are seeded to get the similar appearance and to get a similar texture. That is what scientists are trying to investigate, and soon that will also be made possible. So, moving on, the second topic that was discussed in this week's content is apoptosis, which is a form of cell death. So, again, let's start with a microscopy image shown here at three cells. Let's try to differentiate what we just visibly see from one to another. In this cell here, we can see that there is a lot of vesicles present within it. These are known as blebs. There are lots of bleb formation. And here, the cell on the right, you can see that they have just bursted out. They've just spilled all their uh, contents into the extracellular environment. And here, actually, what you see is a phagocytic cell, might be a macrophage or other phagocytic cells that have internalized another cell, which is presumed to be dead. So, in fact, all of these three images are images of dead cells, but they're so different from each other. In fact, the cells on the left, that is the first two cells, they are apoptotic death, and this is a necrotic form of death. So, in apoptotic death, the cell that dies tries to not initiate a lot of inflammatory phenotype, does not, it tends not to release anything into the extracellular environment, and it kind of dies within itself, and it's later engulfed by a phagocytic cell. So there is no much damage to the surrounding. However, in necrosis, the cell just bursts open, releases all of its content, and this will further trigger inflammation and trigger a lot of recruitment of other cells to this place. And it is known to form necrosis, necrotic form of cell. So let's talk a little bit more about apoptosis and how it is actually regulated in these cells. So actually, the protein family that controls apoptosis is these caspases. There are two types of caspases: initiator caspases and executioner caspases. That within these two families, there are a large number of caspases that are present based on where they act and what they actually what functions they actually perform. So in initiator caspases, what happens is all caspases in general, they have an adapter binding domain and they have a protease domain. These proteases are able to cleave other proteins further down and mediate various functions. So the adapter binding protein will bind to the adapter upon presence of apoptotic signal. Otherwise, this won't be present. Upon the presence of apoptotic signal, there is adapter protein that is present. This will bind to the adapter and dimerize. This dimerize activates the gas phase, and the if you see, the protease domain is kind of reorganized, and it becomes the active center, and it's the center that initiates the cleavage, the protease activity. So this initiator uh, caspases, once activated, will go and perform similar cleavages and similar reorganization in the downstream caspases, the executioner caspases. So now, once all of them are activated, they can go and cleave multiple substrates. They can go and cleave DNA, like they can go and activate endosomes, nucleuses that will cleave the DNA, that will go and destroy the nuclear membrane, cause organelle damage, and so on and so forth, thereby slowly killing the cell. This is known as apoptosis. So this signal is known like as a cascade, right? It starts from one and then goes on multiple proteins. So why cascades exist in biological systems? It is inherently to amplify a signal. So say there is one adaptive protein. It activates one initiator caspase. This one initiator caspase can activate two executionary caspase, which can go and further activate two more and two more and two more. That way, there is an amplification of the signal, and that is what is crucial, and that is why most of the signaling pathways have this cascade formation, including blood clotting. We've all learned about it. Blood clotting also has various factors that activate each other. That is only for amplifying the signal. So there are two types of apoptosis activation. One is an extrinsic activation and one is an intrinsic activation. We look at both of it one after the other. So this shown here is an extrinsic mode of activation. Here what happens is there is a lymphocyte that has a Fas ligand. Your target cell expresses the death receptor that binds to this Fas ligand. So when the contact is established between a killer lymphocyte and the target cell, there is activation of the initiator caspases. Now, this caspases can go and further activate other caspases downstream, ultimately leading to initiation of apoptosis. Then this cell is marked for apoptosis and it will ultimately die. Now, there is an intrinsic mode of activation as well, which does not require the interaction with other cells. Here, what happens is your mitochondria inherently stores a large amount of cytochrome C. This is a protein that is involved in the electron transport chain as well. And this large amount of cytochrome C presence is stored within the mitochondria and is not released into the cytoplasm. However, at certain initiator signals, there is some signal that the cell receives. Various proteins actually control this activity. BCL2 family of proteins majorly control this activity, and because of that, what happens is the cytochrome C is released into the cytoplasm. Here, it can now interact with this protein known as APAF1, ultimately forming something known as the apoptosome. So here, if you see, there is this card domain. Card domain is basically the domain where which multiple units interact with each other and form this multi-protein assembly. To this apoptosome, the card domain of caspases, initiator caspases can interact, and once this interaction happens, it activates the initiator caspases, which then goes on to activate other executionary caspases, and ultimately leading to the cascade and apoptotic cell death. So, what are the characteristic features of apoptosis? Like we saw the characteristic features of senescent cells, there are certain characteristic features of apoptotic cells as well. So, some of the most obvious signals is nuclear condensation and DNA fragmentation. DNA is sheared into multiple short segments. They are not viable anymore. They cannot transcribe or translate any protein anymore. And another classic characteristic of an apoptotic cell is the presence of apoptotic bodies. These are small bodies that may contain damaged uh, damaged organelles, proteins, cytoplasmic contents, and they are released out and they have these eat me signals, which is basically signal for phagocytic cells to come and phagocytose them and then ultimately fuse them with the lysosome, resulting in their degradation. So like this, slowly by slowly, there is this bleb formation on the membrane, which means the membrane is projecting out in the process of releasing out these apoptotic bodies. Ultimately, the entire cell is released out in that way, and the entire cell is internalized by these phagocytic cells. Now, I mentioned that the DNA is fragmented. How this happens is by the activation of nucleases, endonucleases. So in this example, there is the presence of inactive CAD. CAD is the endonuclease, and it is bound to this protein known as ICAD. This is what makes it inactive in the first place. Now, if there is presence of executioner caspases, it will cleave this ICAD domain, making CAD active. Now, this CAD can go and fragment the DNA at multiple locations. Ultimately, here we can see that this is as in when apoptosis progresses. Initially, the entire DNA is present in a single band because they are all organized together, and slowly they are starting to be cleaved. And here we can see that there are presence of smaller bands as known as a smear of DNA, because there are bands of different lengths, and that is why there is multiple bands that are seen. And ultimately, 12 hours post initiation of apoptosis, almost the entire DNA has been cleaved into multiple fragments of shorter lengths, making the cell non-viable again. So this is what apoptosis was. And the third topic that was touched upon in this week's content is cancer. Now, none of us are strangers to cancer. We've all unfortunately heard about it. And the statistics is so shocking and so scary that nearly 1 in every 5 people will die of cancer one day. It's not even about being diagnosed, it is being uh, killed by this disease. So again, like we've done for the previous topics, we will start with this microscopic image. This is an H&E staining. So all the cells have been stained, and this purple uh, stain is the nucleus of the cell. So this is a normal section, and this is a tumor section. Here, the first thing that we can observe is there is a larger number of cells present. Some cells have multiple nuclei, some cells have one nuclei, but they're also closely placed, and there is a invasion of tissue structure. This is a normal tissue structure. They're all organized one next to each other, but here there is just a lump formation. The entire organization of the tissue is lost. So this is a characteristic feature of cancer cells. And if you look at just the overall uh, morphological features of cancer cells, we can see that many cells continue to grow and divide, most oftenly forming a lump-like structure. They are not spaced or organized in an orderly manner. There is variations in size and shape of cells, whereas normal cells, if they are all terminally differentiated, they are all of similar morphology. But in cancer cells, that need not be the case. Um, they are just present in all sizes and shapes. Nucleus is larger and darker than normal. This is mainly because the cell is actively dividing. So the nucleus is in the process of division. Again, the chromosome number that we cannot observe at the outright H&E stain, you need further analysis. But the number of chromosomes is abnormal, the organization is abnormal. In fact, the HeLa cells that we are talking about does not have 46 chromosome, but has greater than 70 chromosomes present in it because of its abnormal division. And then ultimately it forms a cluster without a very organized boundary. So if you actually think about it, your tissues are very organized, right? They have a specific boundary. Your liver does not exceed the boundary of what a liver should be, but cancer cells have no such boundaries. They will form lumps, they will divide anywhere that they have to. So if you think about what a multicellular organism is, it's essentially a sperm cell that is fusing with an egg cell, forming something known as the zygote. The zygote then undergoes multiple rounds of cell division, ultimately forming an embryo that forms specific regions. They are differentiated in different ways that ultimately give rise to a complete organism with different types of cells, different types of tissues organized in a very orderly manner. If you think about a bacterial cell or any unicellular cell, they compete with each other. Every cell has a competition with each other, and they compete to leave behind their own progeny. They compete to leave behind their own DNA. Even though if there are two bacterial cells that are arising from the same parent, essentially they have similar DNA content, but however, they will also compete with each other and will try to gain some selective advantage, some competitive advantage by which they can leave behind greater number of progeny when compared to the others. However, in a multicellular organism, there is a cooperation between cells, even though there are so many billion number of cells in your body, only the gametes will be able to give their genetic material to the future generation. Only their DNA is going to get propagated. The remaining cell is essentially working together so that this can happen. There is mutual cooperation seen, and even though they don't leave behind a progeny of their own, they are existing in this cooperation. So So there are about 10 to the power 14 cells in our body. And even if you consider the number of mutations that occur every day, there are more than billion mutations that will occur in your body on a daily basis. Now, if all of them had to develop into cancer, none of us would be here today. But most of these mutations are harmless. So if you remember, we initially last week spoke about these intergenic regions, which means that the DNA that we actually contain in our cells, most of which are not going to be encoded into any proteins. They are just repetitive fragments. They are just intergenic regions that are present. And if, say, there is a mutation in an intergenic region that is not going to play any regulatory role, not going to have any expression, then these mutations will be harmless. Moreover, we also have a DNA repair pathway. We have very well regulated DNA repair pathways by which these mutations are corrected. So most of these mutations are harmless. However, there are certain mutations that give cells a competitive advantage. They allow the cells to replicate more. They allow the cells to not be uh, an apoptotic body, and stuff like this is what makes a cell cancerous. Now, the image on the left is actually a image of a patient having metastasized cancer, which means it is spread all over the body. And you can see that these fluorescent regions are all cancer cells, and multiple organs have been affected. And this is a terminal stage of cancer. After this, the patient will enter into multi-organ failure and is often not uh, treatable. So if we just look at the overall characteristic of cancer cells, then there are two major differences when compared to a normal cell. One is that they reproduce without any concern for the normal restraints that are actually present on cell growth and division. And they invade and colonize territories normally reserved for other cells. Now, both of these characteristics are very important. So, say this is a duct, any duct, say an intestinal duct, and this is the lumen lined properly with your epithelial cells. Now, say one of the cell becomes cancerous and they divide, convert and form other cells. This is known as a benign tumor. It's not cancerous. It is often treatable by complete removal of tumor, and it will not uh, be fatal to the organism. However, if it starts invading other colonies, then it will ultimately enter your bloodstream, and then it can go to other places and create similar tumors, ultimately resulting in multi organ failure. And this is what kills a patient. This metastasis that we call it is what kills a patient ultimately. So, the cancer starts with mutations, right? So, this process is known as carcinogenesis. It is the heritable mutation that a cancer cell gains, after which it becomes a, a, a rogue cell, rapidly dividing cell that can invade into other species. So the causative agents of carcinogenesis are known as carcinogens, and they can either be chemical carcinogens or radiations. Chemical carcinogens are different type of chemical compounds that may be inhaled, that may be ingested, that may be topically applied, and these chemicals are capable of causing mutations in the local sequence, nucleotide sequence, ultimately down the line causing cancer. So these chemical carcinogens will cause a very long time for cancer to actually develop. Radiation such as X-rays or even the UV light susceptible population can develop cancer because they are also capable of causing DNA damage. So effects of carcinogens are not visible until years later, right? It will take a long amount of time for a carcinogen to actually cause cancer. In fact, even after the Hiroshima and Nagasaki incident, cancer did not start developing in those exposed population of people for at least 5 years after the incident, suggesting that it is not one mutation that causes cancer, but in fact, it is the accumulation of mutation that causes cancer. So say this is the normal epithelial cells and there is one mutant cell, nothing happens. Now the same cell down the line gains another mutation and another mutation. And ultimately has this one mutation that it together makes the cell invasive and completely rogue. So there are two properties in normal cells that the cancer cells seem to defy. This is in regard to replication. It is that the normal cells only divide when they're attached to a surface. This is what I was earlier mentioning. If your tissue culture dish, the cells are adherent. They need to bind to the tissue culture dish, and only then will they start dividing. In suspension, these cells will be unable to divide. However, in cancer cells, that is not necessarily true. They have been shown to divide even in suspension cultures. Now, another property is normal cells do not divide when they reach confluence. Confluence is a term that we use to describe a condition in which your entire dish is covered with cells. There is no more place for the cells to divide. And here they have this property known as contact inhibition, wherein if there is a cell that is interacting with cells on all of its sides, it will not divide, it will be halted in its replicative state. Irreversible, of course, because again, when you passage this, you split them into fresh flasks, they will again start dividing. However, cancer cells have lost this contact inhibition trait, and even if the entire dish is confluent, often they start piling upon each other and they start forming this lump like structure even in vitro cultures. So again, in this microscopic image, this is very clear. These are normal cells. They form a monolayer. They're equally spread. And here you can see that there's just lumps. They have grown on top of each other and overlapping. So what is metastasis? So once a cancer develops, it will invade into the neighboring tissue, ultimately entering into the uh, circulatory system. Once it enters into the circulatory system, they can go and get deposited in a fresher area. So, say this started in your gut and then it entered the capillary, and now it has reached the blood vessel in liver. Now, it can again invade into the liver and then form a new tumor over here. This is known as metastasis. And this is what kills a patient. So, with this, we will start discussing the previous year assignment questions and go through them one by one. So the first question was, which of the following is not a characteristic of cancer cells? The first option is they originate within tissues and grow and divide and diverge further from normalcy. They divide more rapidly than their progenitors. They evade programmed cell death, and that they have a very intricately controlled cell division process. So if you look at the progression of cancer, then it starts with a normal epithelium only. Like I said, the cells start accumulating mutations, and up until that one critical mutation, the cells don't really show any cancerous property. But once that mutation has reached, they start having a development of cancer. But still, here it's just a tumor, it has not invaded into neighboring tissues. But here in the third stage, we could see that it is completely taken over and invaded into neighboring tissues. This epithelial that was once visible is completely lost over here and entirely taken over by cancerous cell. And ultimately, it invades it further into the connective tissue as well. Even though it originated from the epithelium, now it has entered into the connective tissue layer and it will go on and enter into the circulatory system. So, uh, the option that suggested that it arises from a cell, it slowly progresses into its fully blown state is entirely true. Again, there is a property of cancerous cells that they have decreased apoptosis. So it is a normal cell and they have some damage that is accumulated, then they are signaled for apoptosis. They start dying, and again, only the healthy cells are maintained in homeostatic condition. However, in a uh, if there is increased cell division and normal apoptosis, so more than normal the cells are dividing, and even if the some of the cells are killed by apoptosis, there is still a great number of cells that accumulate forming a tumor. Or there is normal cell division and decreased apoptosis, so even then in the end there's a greater fraction of cells that survive, resulting in a tumor formation. So this statement is also correct, which suggests that cancerous cells they evade apoptotic signals. So the only option that does not make sense and it's not correct is that they have an intricately controlled cell division process. In fact, we have seen that they lose any control over cell division. They just rapidly divide and uh, do not have any contact inhibition or do not have any adherence requirement for division. Moving on, the second question was cancer cells may have mutations in which of the following genes. So again, this is a class of genes known as the cancer critical genes. These are genes that are most commonly seen to be mutated in a large number of cancer cells and large number of patients that they have taken sampling from. So there are two types of cancer critical genes: proto-oncogenes and tumor suppressor genes. So, proto-oncogenes are those genes that require an activating mutation, which means that they're expressed in a higher amount, their activity is enhanced, something like that. Some mutation that causes an activating nature, it enables the oncogene to promote cell transformation and ultimately form a cancerous cell. Now, there is also tumor suppressor genes that in its normal function, it functions to suppress tumor. However, when there is an inhibitory mutation, which means there's a loss of function mutation, then it is not able to perform its normal function of tumor suppression and in turn results in formation of cancer. So these two are the type of genes, proto-oncogenes and tumor suppressor genes. So let's look at how proto-oncogenes can become cancerous. So proto-oncogene in its function essentiality does not um, exist in that great of an activity, but however, when its activity or expression level is enhanced, then cancer is initiated. So this can happen by deletion of point mutation and coding sequence. Because of this, what happens is the hyperactive protein is made, but in normal amount. So there is some change in the catalytic activity of the protein, and now it can perform its function better. So even though it's in the normal amount, it is hyperactive. Then there can be greater expression of the protein. So here the regulatory part of the gene is mutated and now it's not controlling it properly. And now because of which the normal protein is present, but it is in a greater amount, because of which again it can perform the function more better than it used to. Or there can be gene amplification, which means that instead of one copy of the gene in the your DNA, it is present in multiple copies, because of which greater amount of RNA is present, greater amount of protein is produced. Again, protein is being overproduced. There can be chromosomal rearrangement. What happens is the sequence of DNA moves to a region where a regulatory sequence comes near to it, or the regulatory sequence moves next to the protein that is encoding, next to the DNA that is encoding this protein, and because of which the regulation of this protein is altered and ultimately causes the protein to be overproduced. Now, there can also be fusion with otherwise overproduced protein. So even though it's fused together, it may or may not be independently translated. And again, there's a greater amount of protein that is produced when compared to normal. So an example for this is cell proliferation genes. So genes that are involved in cell proliferation, if they're present in a normal amount, there is normal cellular proliferation. But if there's greater amount, then there is greater cellular proliferation, or if there is enhanced activity, then there is greater cellular proliferation. So this is an example for proto-oncogene. Now, there are also uh, tumor suppressor genes, and here is an example of the RB gene copy. So this is a retinoblastoma gene copy, and here we need to appreciate the fact that there are two copies. We are a diploid organism, so there are two copies of the same gene present. So in one gene, if it is mutated, then there is still an active presence of another gene that will perform the tumor suppressor activity. So for in order to a tumor suppressor gene to result in a fully blown cancer, both the copies of the gene needs to be mutated, hence making it a very, very rare event for tumor suppressor genes to be the cause of cancer. However, it still happens. For example, this is a retinoblastoma, which requires the mutation of RB gene in both the locus. So, say there is mutation in the maternal chromosome, but there is normal RB in the paternal chromosome. So, in this case, if the paternal chromosome is also mutated by some means, then the cell will become cancerous. So, say there is a loss of this chromosome because of non-disjunction, aneuploidy and polyploidy. Aneuploidy, and because of which what happens is there is only one copy present, and that copy is also mutated. So, this protein is not present to perform its tumor suppressive nature, because of which the cell is cancerous. Otherwise, there is a chromosome loss, first non-disjunction happens, and then there is chromosome duplication. So now instead of the normal paternal chromosome, there are two copies of the mutated mother chromosome, again causing retinoblastoma. Or there is some recombination event that happens, because of which this copy is again lost. There can also be gene conversion, deletion, or point mutation in the paternal chromosome, ultimately resulting in a situation where there is no normal copy of the gene present, which suggests that there is no normal protein that is being present. So this example for this is cell proliferation controlling gene. So if this gene is present, then the cell does not proliferate. But now if both the copies have been mutated, then that gene only is not present, because of which cell proliferation is uncontrolled. So uh, this is an example for one of the most commonly mutated cancer critical gene known as P53. So P53 plays a role in cell cycle arrest, senescence, and apoptosis, which means that it stops the cell from being cancerous. However, there might be certain DNA damage or telomere shortening or other carcinogenic conditions, because of which this active P53 is lost. Now, what happens is it is not able to normally perform its function. It's not able to cause cell cycle arrest, it's not able to cause cell senescence, and not able to initiate apoptosis, because of which the cell becomes cancerous. So, to answer the question, cancer cells may have mutation in which of the following genes? It can have mutation in growth promoting genes, it can have mutation in cell proliferation suppressing genes, and it can also have mutation in apoptosis signaling genes. So, if the cell has a gain of function in growth promoting gene, loss of function in the cell proliferation suppressing gene, and loss of function in the apoptosis signaling gene, there is a development of cancer. The next question is, which of the following is correct about metastasis? So, um, metastasis, again, we have seen previously this very same image, that it is the property of a cancer cell by which it invades into the circulatory system and is able to colonize other tissues, thereby forming new tumors in other regions. So, if you actually see here, then only a small fraction of the tumor enters into the bloodstream. And even after it enters into the bloodstream, there are a large amount of immune cells in the blood and a large amount of killing mechanisms present in the blood, by which it is estimated that fewer than 1 in 1,000 cells that is entering into the bloodstream will ultimately be able to successfully colonize a new tissue. So the chances of these cancer cells succeeding is very, very, very low. And even despite that, metastasis happens. And multiple organs are damaged in this process, because a new location means new organ that the cancer cells can invade into, and the normal cells will not be able to perform their function anymore. So, in order to answer this question, it is one of the terminal stages of cancer, yes, because after that, multi organ damage and the patient can hardly bounce back. Not all cancer cells can metastasize. This is also true, because only a fraction of the cell uh, enter into the bloodstream, and only that can go and finally colonize. A fraction of that can only finally colonize. So, not all cancer cells can successfully metastasize. And during metastasis, cancerous cells enter the bloodstream or the lymphatic system to travel into a new location of the body. So this is the circulatory system. This is what is connecting our entire body essentially. So once it enters into the circulatory system, it can travel to places far away and colonize them. So again, the answer would be all of the above. Pick the incorrect statement about apoptosis. So this question is fairly straightforward. If we read the options only, we will be able to understand. Apoptosis is a mode of cell death to dispose of unwanted cells in the diversity of settings. This is true. Apoptosis involves an orchestrated involvement of multiple members of the caspase family of proteases. This is also uh, true because caspases are the proteases that cleave these executional caspases and then go and cleave other uh, proteins and perform effector functions and thereby help in the progression of apoptosis. And the third option is proteases target random proteins for a restricted proteolysis in an uncontrolled manner. This is not true because proteases have very specific targets and they target these proteins and they perform further effector functions. The final option is apoptosis minimizes damage and disruption of the neighboring cells. This is also true because that is what apoptosis basically is intended for. It minimizes the damage. There are apoptotic bodies that are released that are engulfed by phagocytosis cells and they do not really disrupt the environment that they are in. So the option that is incorrect is proteases target random proteins because again it is very specific proteins that have been targeted. So the next question was pick the correct statement. Senescent cells are never multinucleated. Senescent cells show disrupted nuclear integrity. Senescent cells accumulate dysfunctional ribosomes and display decreased levels of ROS. Senescent cells typically shrink in size. So let's look at one of the image that we had earlier seen. Here cells are typically bigger and they have damaged DNA contents or organization and they have lost the Lamin signal, which means that they have lost the nuclear envelope integrity. So if you with this fact itself, we can see that senescent cells can be multinucleated. They have disrupted nuclear envelope integrity, and are uh, they are not going to have lower levels of ROS and they're typically greater in size. So the correct statement is because these three are wrong. The correct statement is that the senescent cells show disrupted nuclear envelope integrity. Which of the following is an anti-apoptotic protein? So, like I mentioned, there are a large amount of proteins that is controlling the intrinsic mode of activation of apoptosis, and mainly it is from a family of protein known as the BCL2 family. Now, there are different types of proteins within this family only. It can be anti-apoptotic, pro-apoptotic. Anti-apoptotic means that these proteins exist in order to prevent the cell from initiating apoptosis. However, pro-apoptotic proteins exist in order to initiate the apoptotic cascade. So in this question, we have following option: BAX, BCL, XL, BID, and BIC. And we can see that there is this one particular protein, BCL, XL, which is present on the mitochondrial membrane. And upon apoptotic stimulus, they get aggregated together and enhance the secretion of cytochrome C from the intermitochondrial membrane space to the cytoplasm, where it can go and bind to protein and form the apoptosome, as we have seen. However, if there is an anti-apoptotic protein present, then it prevents the interaction of these BCL2 family proteins and thereby does not enhance the secretion of cytochrome. So this anti-apoptotic protein is the BCL uh, XL protein that we've seen, and this prevents the dimerization of the protein known as BAC. So it prevents the dimerization of BAC, which means that cytochrome C is not released into the cytoplasm, which means that apoptosis is not initiated. So the anti-apoptotic protein BCL-XL is an anti-apoptotic protein. Next question is, which of the following is incorrect regarding RB gene? RB gene we've again seen is the gene that is responsible for retinoblastoma, which is a type of cancer. So if it's a healthy individual, even if the individual has a mutation in one copy of the gene, then the individual is fine. They do not develop cancer. However, if there is an individual already having one mutated copy, then it requires only one other mutation in the other copy, ultimately leading to excessive cell proliferation and retinoblastoma. It is also possible that in a normal individual, the chromosomes first gain mutation in one of the locus and then proceeds to gain mutation in the other locus. However, this is very very rare, ultimately leading to excessive cell proliferation and retinoblastoma. So what exactly is the RB gene? So this RB gene codes for a protein known as the RB protein. This RB protein, once active, is bound to E2F, which is a transcription factor. Now, once it's bound to this E2F, then there is inactivation of E2F and the downstream genes are not activated. However, in mitosis, in the G1 phase, the G1 CDK is able to phosphorylate the RB protein. Now, this is inactivated, which means that the bound E2F is now free, active, and can go and synthesize various proteins that is required for the progression to the S phase. So, RB protein is very, very crucial in the progression of the cell cycle, and hence, if this is mutated such that this controlling function is lost, then E2F protein is always active, which means that the cell is continuously proliferating. So the options is it's a cancer suppressor gene. That is true. The phosphorylated form of RB binds to E2F. This is wrong because once phosphorylated, RB becomes inactive. RB is phosphorylated by G1-CDK. This is true. It represses E2F mediated transcription when bound to E2F. This is also true. So the incorrect statement is that the phosphorylated form of RB binds to E2F. Now, moving on, the question states, which of the following statement is false regarding Warburg effect? So again, Warburg effect we have not previously discussed. It is a type of altered metabolism that is seen in cancerous cells. So in a normal cell, how does glucose metabolism happen? Glucose is internalized. Glucose is internalized into the cytoplasm, and here glycolysis happens. With glycolysis, glucose is converted into pyruvate. This pyruvate ultimately enters into the TCA cycle inside the mitochondria, and here it releases through this entire process. Ultimately, it's converted into CO2 and releases a large amount of ATP, NADPH, FADH2, which enters into the electron transport chain, which uses O2 as the primary electron carrier and ultimately forming water. In this process, known as oxidative phosphorylation, because O2 serves as the initial electron carrier, there are about 38 molecules of ATP produced and entire conversion of glucose into CO2. However, in the absence of oxygen, what the cell does is it undergoes anaerobic glycolysis, in which the pyruvate formed is reformed into lactate in order to regenerate NADH that is required for glycolysis. Moving on. So this has very low amount of ATP generated, but it's very quick and it's a mode of respiration in anaerobic conditions. So in a cancerous cell, it has been observed that this conversion of pyruvate to lactate is active even in the presence of oxygen. So normally the cell does not prefer this, because if you can see here 38 molecules of ATP is produced and here only 2 molecules of ATP is produced. So there is a very large energy gap and it's not efficient at all. However, a cancer cell only requires energy quickly and also requires carbon source for biomass incorporation. So to facilitate this, there is a Warburg effect that occurs in cancer cells, which has anaerobic glycolysis. One, there is a greater amount of glucose being transported into the cell and glycolysis happening, a greater amount of pyruvate being formed, but very low amount of pyruvate enters aerobic respiration. Most of it is converted into lactate, and 2 molecule per glucose molecule, 2 molecule of ATP per glucose molecule is generated, and ultimately this lactate is used to used as a source for biomass, source of carbon for protein synthesis, for synthesis of essential proteins that will help sustain the cellular proliferation that is seen by cancer cells. So if you look at it, then a differentiated tissue has glucose, glycolysis, pyruvate, oxidative phosphorylation, and very low amount of lactate. However, in the case of tumor or a proliferative tissue, so tumor cells are more uh, comparable to embryonic cells, which also have these Warburg effects in them. So in these cells, if you think about it, glucose is actively converted into pyruvate, and large amount of this pyruvate enters into the lactate anaerobic respiration. So here lactate produces low amount of ATP but regenerates the NADPH that is required for glycolysis again. This produces building blocks that can be used by the cell to generate other proteins and other enzymes that are required for their normal functioning. So if you look at these statements in order to identify the statement that is false, first it is the hallmark of cancer. Yes, there is a preference to metabolize glucose anaerobically under aerobic conditions. This is true. There is a preference to metabolize glucose anaerobically under anaerobic conditions. This is false, however, because anaerobic condition does not exist normally in your cell. You are still breathing. The cells still receive a large amount of oxygen, but still actively chooses to metabolize it anaerobically. Warburg effect leads to an increase in production of lactate. This is also true. So the statement that is false is that anaerobic respiration happens under anaerobic condition. That is why there is a term given to this Warburg effect, which is anaerobic glycolysis. So this is when even in anaerobic condition, even in aerobic condition, anaerobic respiration is taking place. Moving on, the question is caspases belong to which class of proteases? So we've seen that caspases are proteases that play a very crucial role in apoptosis. Here we can see that the name caspases originated because they were named after their core function, cysteine aspartic proteases or cysteine aspartases or cysteine dependent aspartate directed proteases. So, because of this, the answer is fairly straightforward. They are cysteine proteases. The final question is, which of the following proteins increase aging? So again, we've seen that aging, senescence, they are all very tightly regulated and only is initiated upon damage accumulated in the cells. And even though this is a very complicated, seems to be a very complicated representation, let's focus on one thing. So mTOR is a complex that is present in the cell and it controls various uh, uh, functions of the cell, including inhibition of autophagy. So if mTOR is very active, then there is inhibition of autophagy, which inhibits the increase in lifespan because there is no clearance of damaged organelles, no clearance of damaged proteins, and that gets keep on accumulating, triggering senescence faster. Now, again, if mTOR is more active, then there is increased cell cycle, increased protein synthesis, and ultimately leading to increased inflammation. Again, inflammation is not very favorable for increased lifespan, and this will reduce the lifespan. There are also production of these plaques, these tau plaques that are commonly seen in Alzheimer patients, or necrosis that again is a necrotic form of cell death that we saw as just a burst cell death, again resulting in inflammation, ultimately causing a reduction of lifespan. So in this question, the answer is mTOR, and it is a protein that regulates multiple pathways of the cell. However, when it is activated, then the cells start accumulating damaged organelles and damaged proteins, ultimately leading to increased aging. So that is all from my side. Let us discuss. I know that you have an exam very, I think this weekend or something. So if you have registered for the exam, you might have some questions. So we can discuss before we end this session.
Hi, ma'am. So, I have a general doubt. Like, are you uploading these videos in YouTube or any other platforms?
Yes, these videos have been uploaded on YouTube. I think I shared a link in the last class on this chat session only. I will reshare this link.
Okay, ma'am.
So I pinned the link for the YouTube video. That video is one of the sessions, but you will find all the sessions within the same playlist. So you can go and take a look at that. All the 7 sessions, even today's video, I will upload in the same place. So all the 7 videos will be present there. Is there any other thing that you would like to discuss before we end this session? Okay, then thank you all for attending. All the best for your exams. I think there'll be one final session next week wherein we can maybe discuss if you have any questions in the exam that you appeared for. Otherwise, all the best. Hope all of you do well. Have a nice day. We'll end this session here only. If you have anything, you can write to me or you can post in the discussion forum of the NPTEL course in itself. We will get back to you.